Well, good evening, everyone. I'm Peter deSilva, Board Chair of the CMTRF, and welcome to another installment of the CMTRF Scientific Update webinar series. Get ready. We have an amazing program this evening. You're going to hear from two companies working exclusively in CMT. One will discuss their upcoming phase 2 clinical trial for CMT, another incredibly exciting moment for CMT research. And the other will talk about a new CMT program that is just getting underway. You know, we know at the CMTRF that one trial is not going to end CMT. It's going to take dozens, maybe hundreds of trials to find the right solution for all types of CMT. And I got to tell you, as a patient, it can't happen fast enough for those of us who suffer from this debilitating disease. Before we get going this evening, maybe just a little bit of housekeeping. We're certainly going to reserve time at the end for questions. And so please, as you think of these questions, please type them into the Q&A feature on your screen. Susan Rudiger, CMTRF co-founder and chief mission officer, will moderate the questions after the talks. To the extent there are unanswered questions, we'll certainly do our best to get those answered to you. Now, before we hear from our experts, and we do have true experts with us tonight, allow me to share just a bit of my personal background with CMT. So I have CMT1A, as do most of my family members. My two brothers, my sister, who lives her life in a wheelchair and has for 25 years, nieces, nephews, and many other extended family members suffer this dreaded disease. I can still remember the day when my doctors in Boston told me when I was a teenager having my first reconstructive surgeries that I shouldn't really worry because there was about to be a cure for CMT. And he said, certainly in your lifetime. That was 50 years ago. 50 years ago, I was promised that there'd be treatments and cures for CMT. And yet we sit here today without a single treatment or cure. I don't know about you all, but 50 years is long enough. The time is now. We need to get this solved. You know, when I joined the foundation board a few years back, there was conversation already brewing about how to attack CMT given the variety of types and different manifestations. Leaders from the CMT Research Foundation began asking themselves if there was only one type to initially focus on, not exclusively, but initially focus on, the one that might get a solution fastest, which type would it be? Well, the answer was unequivocally CMT1A. So you might ask why. This was a smart, science-based decision, which is how we make all decisions at the CMTRF. We knew that the science was the furthest along in 1A. The basic science was there. We understood that the duplication of PMP22 was the root cause of the issue. We could see a pathway to treatment and ultimately cures. And due to its prevalence, it was really had the most interest from the pharmaceutical industry. But the prevalence was really less important to us. It was the science. We followed the science. Quite frankly, we just knew more about 1A than we did any other type. And that probably still remains the case today. So with that in mind, in September of 2021, the foundation launched ENDGAME, a bold, audacious campaign to raise $10 million to get after CMT1A. Never been done before. No one had tried to raise $10 million to focus on a single type of CMT. Well, I'm proud to say today that we're not done, but we've raised $8 million, or 80% of the $10 million that we set out to raise. We feel good about that, but it's just not good enough. We want to finish this campaign this year. We promised the board we'd do it in 3 years. That 3 years is up in September of 2024. And so we got a little bit more work to do, and we just need to pedal faster and harder to get it done. I certainly hope anyone on this call who's motivated to do so will help us meet that goal after listening to the great scientists we have with us tonight. So with that, I'd like to turn it over to our CEO, Clary Simpson, to discuss how we've deployed the money that we've raised thus far. Clary, over to you.
Thanks, Peter. It's wonderful to see all of you here tonight. Thank you for coming and for being here and taking some time to spend with us. You know, to date, the foundation has funded a total of 12 CMT1A projects. And my job tonight is to give you a very brief snapshot overview of those projects. So these 7 projects focus on developing treatments for the root cause of CMT1A. As Peter said, we all know it's the duplication of PMP22. But if you can't get the treatment to where the damage is happening, it's not going to be effective. So all 7 of these projects are focused on both developing a treatment and ensuring that its delivery to the peripheral nerve system, and specifically the Schwann cells in the PNS. 2 more projects, which you'll see down here on the bottom, focus only on optimizing delivery vehicles for improved delivery, and they can carry different treatment payloads, including those for multiple types of CMT. These projects are at Emory and at Nationwide Children's. And these 3 projects that you see in front of you now are focused on attacking nerve degeneration. While it's not a root cause of CMT1A, nerve degeneration very surely causes CMT symptoms. So we believe that these approaches may have the therapeutic effects of stopping or even in some cases reversing the progression of symptoms of the disease. And then some of the funds go to the costs of scouting, negotiating, and managing the projects. Now, of the 23 companies around the world that are currently working in CMT1A, 10 were new to the field and have partnered with the CMT Research Foundation over the last 4 years. We've almost doubled the number of companies now working on treatments for CMT, and we're very, very proud and excited about that. 2 of the early CMT-funded projects have resulted in acquisition or licensing deals that will carry the assets forward. As many of you know, DTX Pharma was acquired by Novartis last year, and Cleopa's project out of Cyprus was licensed by Armatus Bio. Recently, Augustine Therapeutics has secured significant funding to take their CMT asset forward, including an investment from Lilly Pharmaceuticals. And also, 5 of the assets from funded projects are now preparing for clinical trials. We also have 3 academic centers focused on CMT1A that surprisingly were not working in CMT before ENDGAME started. Nationwide Children's Hospital at Ohio State University, Emory University in Atlanta, and Vanderbilt University. So we have great new partners. At the CMT Research Foundation, we know that scientific discovery is about continuous improvement and innovation, and drug development is about creating treatment options that work for all patients. Many of you have heard us say many times, we need lots of shots on goal. So this summer, we have 3 new projects that have been enabled by your generosity. The first is Alloy Therapeutics. It's a project based on splicing the RNA to silence the message of too much PMP. Dr. Paul August is the PI on this project. He's a current scientific advisory board member and a former board member of CMT Research Foundation. The next is a project that we have not officially announced, so I'm secretly telling all of you this now. Dr. Afrouz Rashnanajad at Nationwide Children's Hospital has a project that aims to develop new vehicles for delivering gene therapies more efficiently into peripheral nerves to improve treatment efficacy and safety of gene therapies for several different types of CMT. So we know that patients need a variety of treatment options, and we're exploring numerous delivery vehicles. Dr. Rashnanajad, for those who don't know her, is widely known as an AAV expert internationally. She started her own lab with work on CMT1B, which was funded by the CMT Research Foundation, and now she's applying her focus on CMT1A. And last, but very much not least, is 82VS, a division of Alloy Therapeutics. It's a terrific new partnership that we're developing with 82VS and Dr. Carlos Loya. This work will explore whether antibodies can deliver ASOs, antisense oligonucleotides, possibly better than Ionis or even DTX. Our scientific advisory board think that this could be the next best shot for CMT1A treatments. So you might be interested that we met the 82VS team at Bio 2023, and we introduced them to the unmet need in CMT. So we're very happy now about this developing partnership. You will hear more about their project shortly from Dr. Loya. We are very, very excited, as I hope you all are, about upcoming trials for 1A. We maintain regular communication with Novartis and look forward to an announcement in the near future. But today, you're going to hear from NMD Pharma about their plans to start recruiting for phase 2 trials in CMT soon. In September of 2024, if everything goes as planned. So here to tell us more about that phase 2 trial is Dr. Jorge Quiroz, the Chief Medical Officer at NMD Pharma. Dr. Quiroz?
Good afternoon. Thank you, Clary, for the nice introduction. I'm happy to share with you what we have been doing at NMD Pharma and what we're planning to do, hopefully getting into the clinic in the next couple of months, as you will hear in a second. I would like to confirm that the screen is being shared adequately. Great. So NMD Pharma is a small biotechnological company founded 7 or 8 years ago in Denmark. And now the company is present in Denmark and in the US. The major investors of the company are another pharmaceutical companies under venture funds, Roche, Lundbeck Foundation, Novo Nordisk, Incef, and Jato, most of them from Europe. What we're doing at NMD Pharma is to focus on the development of drugs that are inhibitors of the chloride channel, the type 1. So it's a chloride channel type 1 inhibitor. We call our drug, the one that is on the clinic, NMD 670, and you will hear that name later on. As you know, earlier on during drug development, a lot of numbers that help us to keep track of our drug candidates. The action of this drug is to inhibit this chloride channel that is present all across the muscle. As you know very well, the impulse goes from the brain to the spinal cord, and from the spinal cord through the motor neurons to the muscle, and that's where the CMT disease finds its pathophysiological organ. This drug acts on the muscle. It's beyond the effects that one may have or not in the synapse or in the axons or the Schwann cells of these nerves. We believe, for the reasons that I will share with you in a second, that will be a very good candidate to try in the disease, and I will share with you the rationale. We are developing this medication for neuromuscular diseases in general. We have 2 ongoing trials with this drug, the NMD 670 that I just mentioned, in myasthenia gravis, and the trial has started a month ago, and is already recruiting patients, and also in spinal muscular atrophy, certain subtypes, and in adult patients, and that study is ongoing. We are close to halfway through, started last year, and very happy with what we have been observing in terms of the profile of adverse events so far. And the third indication, and we just have an IND opening, which is the authorization that the FDA gives the companies to start trials in the US, was received by us from the FDA, and we're in the latest stages of the approval for Europe. With all of that, we expect that we can start the enrollment of this study, and I will share with you the details of the study as well, in September this year, but with the caveat that that takes into account that all the contracts with the sites are elaborated and signed, and that also all the ethic approvals have been received. So if everything goes well, we expect that that will be the initiation date, but bear with us if that is not the case and we slip into the 4th quarter this year. In all cases, it's a couple of months away. This drug, NMD 670, let me move something from my screen. It's a drug that, as I mentioned, acts on the neuromuscular junction and in the muscle. As you can see, and let me see if I can grab. As you can see, this is a picture of the neurotransmission between the axon arriving to the muscle in the red color, And you can see the receptors of acetylcholine, which are the ones that when stimulated by the acetylcholine release in the motor neuron, produce the muscle contraction. The chloride channel 1, the one that these drugs act on, is a channel that you can also find in the terminals and all along the muscle fiber. And the action in the physiologically speaking, of this channel is to counteract the excessive activity of the muscles. And that is depicted here with a positive sign for acetylcholine and a negative sign or a division sign for CLC1. So what this drug does is inhibit that chloride channel, helping the muscle cells to be more excitable for any amount of acetylcholine that the muscle receives. So we expect that in diseases where the balance for contraction is not optimal, this drug may help to increase the muscle strength and function. So in fact, we have conducted a proof of mechanism study in patients with myasthenia gravis, and we just published this study a couple of months ago in the journal Science Translational Medicine, where we tell the story of the mechanism of action of CLC1, the one that I have shared with you, but also the results in a group of 12 patients with myasthenia gravis, which is a different disease, but the important piece of that information is that it helped acutely after one administration to increase the strength of the muscles and increase the function of the muscles as measured by different endpoints. So with that in hand, we wanted to understand if that was also to be found, that a positive effect in patients with Charcot-Marie-Tooth. As mentioned, myasthenia gravis, which is a disease that we categorize as post-synaptic because the defect is mostly, in most cases, on the opposite side of the receptor to the one from the motor neuron, is different from the diseases that are called presynaptic, like, for example, Charcot-Marie-Tooth, where the defect is in the axon because of the degeneration of the axon or the demyelination of the nerve. But also there are other diseases where these defects are found and where we believe, and we have some evidence now with preclinical information that is telling us that the inhibition of this chloride channel will help to increase this muscle strength and function. And these are some of those diseases. And as I mentioned to you, we are already running studies in the 3 ones that are blacked in this slide. So in sum, the CLC1 inhibitor, NMD 670, enhances muscle excitability and restores neuromuscular transmission, regardless of whether this function is presynaptic or postsynaptic. There are ways to understand these deficits from the electrophysiology, and some of you may have gone through these examinations. There are 2 ways to measure how the neuromuscular junction, which is the place where the neuron meets the muscle, is functioning. And some of them are measuring the electrophysiology, which is the electrical activity of the cells in those muscles, which is the single fiber electromyography. Or you can measure the muscle in its totality through measurements called repetitive net stimulation. And those are telling us the type of defects that you can find in that neuromuscular function. So in a nutshell, and not to be very deep on this, but happy to answer questions that you may have later, the normal function is that the 2 pulses or more are following one another, but when there are problems in the neuromuscular junction, you can have a delay of one impulse versus the first electric impulse, or you can have a block of that second impulse, so a delay or a block, and that in technical terms is called jitter or blocking. Or when you're measuring the entire muscle electrical activity, you can see a decrement in the amplitude of several trends of contractions or fiber activations that you can measure electrophysiologically, and that is what we call the decrement. So you can measure the deficit in neuromuscular function with these 2 mechanisms. As you know, and I will not stay in this slide longer, the disease is not properly a disease of the neuromuscular junction if one were to think de novo. But if you can think again, of course, in the normal condition, the neuromuscular transmission is very relevant to be intact. And of course, when there is demyelinization, the electrophysiological activity of the neuromuscular junction, it gets decreased, and which is more probably accentuated, and we'll show you data about that in the case of the axonal degeneration, as in the CMT type 2 disease. So we wanted to understand, would the patients with CMT disease have these neuromuscular junction transmission deficits? So for that, we conducted an observational study in Denmark and in the US in patients with Charcot-Marie-Tooth disease. And we wanted to understand if there is a deficit in the neuromuscular junction, and for that we use the electrophysiological tools that I just mentioned. And also we wanted to understand, if that were to be positive, how that could translate into a very well prepared or designed phase 2 study, which is what we're going to do next. What are the clinical tests are applicable, and share that information with the agencies and receive their feedback. So in this observational study, we were able to investigate the neuromuscular transmission and also clinical outcome measures for CMT in 21 patients with CMT and in 10 healthy controls. So we were able to compare the measurements, electrophysiological deficits, and the activity, clinical outcome measures, in controls versus patients. And this is what is called a phase 0 study, which is an observational study. No intervention is given to patients. That is no drug involved in this study, just a natural history study, which is for us to investigate if these parameters were altered in the patients or not, and therefore if a drug that will improve that neuromuscular function, this regulation, will be appropriate for patients with CMT. This study was conducted, as mentioned, in Denmark by Professor Henning Andersen, and at Ohio State University by Professor Dave Arnold. And you can see here the list of tests that we were trying to understand if were doable across sites in an international environment for patients with CMT. And very importantly, I don't have more slides with this level of data, but please bear with me for this 1 second. We found that when you compare the healthy controls that are in gray with the patients with CMT that are in green, there is a much larger deficit on the jitter, which is this delay in the train of contractions, or with the blocking, which is the absence of the contraction in train stimuli for patients. So we were able to demonstrate something that was known preclinically, but this is the first time that I was observing patients, that patients do have neuromuscular junction deficits. That opens the possibility that if there is a drug that were to improve that neuromuscular junction deficit, would that improve the symptoms? So the second question that we were able to answer is that this the degree of impairment measured as jitter or blocking, and here you see in the table, correlates very nicely with the degree of symptomatology that the patients were having, as measured by these outcome measurements that some of you may know well. Some of them are the manual muscle test and how much you can flex or not certain muscle groups. But as you can see here, all the other tests that are well known for the community, the 6-minute walk test, the type up and go, the 9-hole peg test, etc. So what these numbers represent is a correlation between the degree of symptoms and the degree of neuromuscular junction deficits. So with this data, I will not show you about this, but we back translate into animals with models with CMT, and we have positive results. We do believe that the inhibition of this channel, the chloride channel 1, may result in an improvement of the neuromuscular deficit, and therefore an improvement on the muscle function and strength. So with that in mind, we presented this data to the agencies in the US, and as you heard earlier, in Europe, and we got an approval to conduct this study. It's a proof of mechanism study, or a phase 2A study in these countries. This is a randomized, double-blind, placebo-controlled study that will evaluate the efficacy, the safety, and the tolerability of NMD670, our chloride channel inhibitor, over 21 days of treatment in patients with CMT disease, the type 1, type 2, but that are ambulatory and adults. We believe that this mechanism of action will be also helpful for younger patients or for non-ambulatory patients or patients with higher difficulties or larger difficulties on mobility. However, for this proof of mechanism study, we're focusing on this very narrow set in order to be able to dissect very well the results that can be obtained after thoroughly investigating NMD670 in patients. And again, the objectives are to investigate efficacy and safety, and the design is the one that is mentioned on the title. Patients are going to be screened and then blindly randomized to receive placebo or to receive NMD670 400 milligrams BID. This is twice a day, one at 8 a.m., the other one at 2 or 3 p.m. And half of the patients will receive one intervention and half of the patients will receive the other. And after that, we'll follow up the patients for 7 days. Because we know that there are much more patients with CMT1, but we want to understand if this is also efficacious in CMT2, we're doing something called an stratification, which is to reserve certain spots for patients with the type 2 disease. We expect to hopefully enroll approximately 30% of the patients, of these 80 patients in total, having the disease being of the type 2, to enable us to reach broader conclusions about the applicability of this information and this data into even other populations with CMT. We will have approximately 20 sites in the US and Europe, and we commented about the date. I'll tell you a bit more details on the next slide about the study and the components. We have inclusion criteria and exclusion criteria, and I have listed here the most relevant ones, and we can discuss them during the Q&A if you have decided to do so. There will be male or female patients and needs to be adults. So we determined that needs to be more than 18 years old, and that because of regulatory mandates, but not older than 70 years old for this first study. They need to walk in the 6-minute walk test at distance that is 1 standard deviation from the mean, but that age and that gender by compared to norms that we have developed. So I understand that it's a bit obscure in terms of how many meters, because for each male or female, the amount to be walked is different. And for each age, that distance also changes. So according to those parameters, age and gender, we are going to determine if the patient is eligible or not. Patients need to have some disease. The manual muscle test for the ankle dorsiflexion is to have a strength of 4. And if you're familiar with this score, it's something that the physicians do when you go to the clinic to see how much you can dorsiflect the foot or not against gravity or just passively. 5 means that there is very little disease or none. 4 means that you have some disease, but we can go all the way to 0 given the other inclusion criteria being met. We have a restriction on the body mass index, but it's very wide. This is patients with normal weight or a little bit perhaps underweight, and patients with overweight and even more than overweight with an index of 35. That will be determined at the sites when you have the visit, and as you know, it's calculated by the height and the weight. And there are other inclusion criteria, but the idea with the medication is that if patients are taking medications, they can continue those medications as far as they are stable during the trial, and doses does not need to be changed, because if not, as in any study, you will have 2 variables to get conclusions on that will make it very difficult to interpret. So we have to ask the patient to be relatively stable in the disease and with the use of medications. On the exclusion criteria, on the other hand, any other disease that may interfere with the interpretation of the data will have to be excluded, and that will be evaluated with the investigator, so the physician that will be evaluating the participation of those patients. We have also established that if you, for this particular study, if you are using AFOs, you will not be able to participate. The inserts are allowed, but other AFOs are not, and that is solely for reasons of this proof of mechanism study. We believe that the drug will help with or without AFOs, but to investigate the research question, we have focused and narrowed the population in this way. Also, if there is a severe deformity or an ankle contracture, then in the opinion of the investigator, will not allow the patients to complete the test, and the tests are very similar to the ones that we have discussed for the phase 0 study. I'll tell you more about that in the next slide. And also, if a patient have had recently ankle surgery or other limb surgery procedure related to CMT, we need to wait for 9 months to complete recovery before the screening, to avoid by the same token that the main inclusion criteria to have different variables occurring at the same time during the clinical study. And this study have the objectives that we already mentioned, efficacy and safety, but here I wanted to give you a more detailed view of what those objectives are. So for the primary endpoint, which is the way that the studies are structured to obtain meaningful responses, we are planning to have the 6-minute walk test as our primary endpoint, followed by the 10 minutes walk run test that you may know well. And on the last place, but also on the primary endpoints, the time up and go for the drug. This will be measured compared to baseline after 21 days and compare the group receiving drug versus the group receiving placebo. And remember, this will be blinded for everybody. Double blinding means that the investigators will not know your allocation to medications, and the patients will also not know to which group they were randomized, if drug or placebo. We're measuring other scales that have been utilized in the recent literature to investigate the effect of drugs in CMT. And as you mentioned as you heard in the introduction, unfortunately not successfully. So the CMT foam is one of them. We're going to have the total score and the individual items. A fatigue index, that is something that we use to understand the performance during the 6-minute walk test. The ONLS score, the CMT high, which is a health index related to the CMT foam and developed by the same group in Rochester. Another reported patient outcome is called SF36, the total score and the individual domains, and also electrophysiology to understand if we're doing what we think we need to be doing with the drug, and that's jitter and blocking. Of course, we're going to do the physical examinations before, during, and after. We need to have some ophthalmical examinations and several laboratory parameters, as in any other studies, before, during, and after, precisely to prove that there are no important adverse events associated with the investigative drug. So with that, I think I will finish. These are the sites that we're planning to have in the study. This is not final, so I emphasize the word potentially participate. We are in conversation with all of these sites, some of them more advanced, some of them less advanced, some of them already finalized in the conversation before the approvals by the agencies and the ethics committees. So we're going to do, you may know, the physicians in Harvard, at Rochester with the Herman, the group that is also the CMT foam, Jeff Sutland, Thomas Brannagan, Bakri El-Sheikh, which is in Ohio, and Dave Arnold, that was in Ohio and now is in Missouri, Gregory Carter, and Yesar Hussain in Austin. It goes without saying that none of this could have been done without the help of the patients and families that were able to participate in that phase 0 study that is enabling us to do a very well-informed, I think that hopefully the best informed trial in CMT so far. To the physicians that perform those investigations, of Dr. Anderson and Dr. Arnold, and the list of colleagues, which is my group at the NMD Pharma, that help us to prepare the clinical trial and put it all up and running. So thank you for your patience to have listened. I'm very happy to answer questions towards the end of the hour.
Thank you so much, Dr. Quiroz. It's really wonderful. This is great progress. I know us in the CMT community, we're so happy to see the inclusion criteria and the sites. And I know we're going to have a lot of questions at the end For those of you who've been raising your hand throughout the webinar, just type your question in the Q&A and that will give you the opportunity to ask it. I'll ask them at the end. So thank you for this information. It was just wonderful, and look forward to the Q&A session. So obviously the momentum for CMT drug development is totally palpable. I have to tell you, I'm Susan Rudiger. I'm the co-founder of the CMT Research Foundation. And in 2018, when we attended our first bio meeting, and that's where we do our speed dating with the other biotech companies, I was working so hard to get interest in CMT. And I asked for 1 million meetings, and I only had a few that were responded to, and the interest was pretty low. This year, when Cleary and Dr. Agusquiza, our director of research, went, they had 35 meetings that they accepted. They turned down many meetings because they had the opportunity to be really selective, and they had to be selective with their time. So it's acquisitions like DTX and announcements like this from NMD and licensing deals by Armidas that are building the momentum for CMT research. I think we can all be really proud of the progress that's being made. But I want to tell you a little bit about a new partnership that we're establishing with Dr. Carlos Loya and his team at 82VS. As Cleary mentioned earlier, we met them at Bio 2023. They had never heard of CMT before, a little convincing on our part, and we were able to get them to come to our convention, which is every year in September in Cambridge. And so Dr. Loya came to our convention in 2023, shortly thereafter submitted a proposal for research, and our scientific advisory team was so excited about the proposal and about the potential for the application of their technology to CMT that we're working on establishing a partnership to get this project going. So I would like to introduce to you Dr. Carlos Loya, who is here tonight to tell you about the newest CMT project underway. Dr. Loya is a neuroscientist by training, and he is a biotech builder. We're going to hear from him in a moment. Thank you, Dr. Loya, for being here.
Oh, thank you, Susan. It is my distinct honor to be invited and really to speak with you tonight. So just to give you a bit of background, so I'm a venture partner at the 82VS Venture Studio, which is a Boston-based biotech venture group that is part of Alloy Therapeutics. Just also as a little bit of background, I'm a neuroscientist by training. My real passion is to really understand the brain and to develop life-changing medicines for neurological diseases. And, I've had the privilege over the last 15 years to help build biotech companies and that have brought many medicines to the clinic, and 2 that are currently FDA approved. And well, my objective for tonight is to just briefly share with you an overview of the delivery technology that we're excited to work with the CMTRF with the goal to develop medicines for CMT1A and many other types. So again, thank you, Susan and the CMTRF for this invitation. And really, as Susan and Cleary mentioned, for raising our awareness of CMT, the unmet needs within the community, and really inspired us, myself and many of my team members here, to think about how we can utilize our drug development and discovery technologies and make medicines for the CMT1A community. So next slide. So just briefly, our vision at 82VS really is to, one, build the most capital efficient biotech companies in the world, because we believe that this will lead to more drugs and solutions for patients ultimately. And we do that by using the drug discovery platforms and R&D services of Alloy Therapeutics. I know this is a general audience, so I really will not kind of go into too much jargon about drug discovery platforms and what we do, but really what Alloy is a biotech company that has deep expertise in generating novel drug discovery platforms or ways to make new drugs. And these platforms and technologies really are made of largely 2 things. One, antibodies or antisense oligonucleotides, what we call genetic medicines. And really the power of the antibody platforms really is to is that they can very specifically bind to proteins outside of a cell that are causing a disease, or bind to proteins that help deliver drugs to a specific cell of interest, like acting as a shuttle or delivery truck. The power of antisense oligonucleotides, or the genetic medicines that we're developing here, is that they can target genes inside of a cell and correct or partially correct the disease-causing effect. So a little bit more about Alloy and what we've been able to do is that, well, Alloy has worked with now over 150 pharma and biotech companies to make medicines, and since 2019 has helped partners in this very short period of time bring about 9 different antibody drugs into the clinic. So it's one of our kind of passions and missions to democratize our technologies and help make medicines together with other partners. So kind of, again, with speaking with Ryan, with Susan, and the experts of the CMTRF team, this made us aware of the current challenges in CMT therapeutics. And as Cleary mentioned, one of the major ones being delivery of medicines to treat CMT. So this made us think of maybe bringing 2 of our tools in our arsenal of drug technologies into play, and one being the antibodies that can help deliver medicines to specific tissues, and 2 are antisense oligonucleotide therapeutics that can correct the effects of a disease-causing gene. So I like to think of kind of the analogy when using these 2 different tools as kind of the antibody being a delivery truck that helps you, take the antisense drug, this being the package, to your home, which is a cell type where we want to correct a specific disease-causing gene effect. So next slide. Really, what and this kind of is what I'm calling right now kind of a next generation of delivery technologies for CMT, and kind of we're part of kind of this next generation, which Cleary alluded to earlier. One of these next-gen delivery technologies is fatty acids, as being kind of a novel type of delivery truck that can take the package, the drug, into cells that are kind of fatty acid loving or lipophilic, and deliver kind of the drug that's going to knock down or bring down PMP22, which is overexpressed, as many of you know, in CMT1A. Another way is utilizing what is called an AAV, or adeno-associated virus, which can take kind of the drug inside the viral particle and deliver that into the cell, and also enable the lowering or the knockdown of PMP22. And our approach, which I'll just go into kind of an overview in the next slide, is we're leveraging kind of this antibodies, this very specific types of drugs that will be targeting a specific receptor in that can be found in the cell surface of tissues that are relevant to CMT, and deliver the drug, which in this case would be an antisense, to knock down PMP22. So next slide. Really, we believe really the CMTRF kind of being an ideal partner here to help us develop this, what we're calling antibody oligonucleotide conjugates, or AOCs, and they're made of this 2 different parts. So we have, proprietary technologies for both the antisense oligonucleotides, which we call anticlastics, and these we've seen, and have validated in multiple different target types that have enhanced potency, stability, less pro-inflammatory responses, which means better safety. And we believe that we can, target here genes that are relevant to CMT with this technology. And then we can leverage a next generation delivery approach. So we've also developed here antibodies for TFR1, and we have developed them with the profile that is, we believe to be very relevant and amenable for for delivering medicines into the peripheral nerve of CMT patients. And the main, I think, advantage of this technology is one, that we can systemically administer this drug. So it would be an intravitreal delivery of this drug, and the antibody or the delivery truck will take the drug into kind of the peripheral nerve of interest, and then knock down the PMP22 or other genes that are relevant to potentially other CMT types. And ultimately, the goal is to develop what we can potentially say is a superior treatment for CMT1A. So I think, as I mentioned earlier, this is really an honor to being invited to speak here today and introduce kind of an overview of what we've been discussing with the CMTRF, and really thank Susan, Ryan, and the team for really introducing us to this community. So I'll stop there and thank you.
All right. Well, thank you, Dr. Loya, and thank you, Dr. Quiroz, for those stimulating and exciting talks. I mean, what a difference. What a change. Just in a few years, so many more scientists are aware of CMT and working in this space. It's quite remarkable. I have to say that I don't know exactly when the first therapies will happen, but I'm confident, I can say this with extreme confidence, it won't take another 50 years before we get those therapies for sure. So thank you guys for your personal and professional dedication to finding therapies for CMT. Our job at the CMTRF is to, look at the science, look at where some of the greatest advancements are being made, and then put our donor donations immediately work. And you know what? That's what we do. We've raised a lot of money. We talked earlier about raising $8 million. None of it's sitting in a bank account. It's basically all been deployed against some of the best science we can find anywhere on the planet. Many of the 35 companies Susan's referenced that they met at BIO this year, they want to move forward with more CMT1A projects. That can't happen without funding. So we need to continue our fundraising efforts to ensure that we have a solid pipeline of potential drug therapies coming through the pipeline. One of the things I was reflecting on is how ENDGAME, this $10 million campaign, has really increased interest in CMT just generally. In 2018, as Susan said, she was walking around these conferences aimlessly, probably, trying to find somebody to talk to. Now, I don't want to say they swarm to her, but they definitely seek her out. They seek her out, they seek Cleary out, because the world has changed and CMT is becoming an actionable issue to be solved. The DTX acquisition by Nevada certainly helped in that regard. It really helped to really extend the value proposition for those working in the peripheral nervous system. Pharma is more interested than ever, and that's a good sign. Again, I'm confident treatments and therapies are on the way. Looking ahead, our internal scientific team is led by Dr. Ryan Igosita, our Director of Research. Ryan, do you want to say hello? Is she out there? Hello, Ryan. We got her picture up there.
Hello.
Hey there. How you doing?
Nice to see everyone.
It has been an amazing addition for us to have Ryan on the team. She leads our advisory council, the scientific advisory board, and she's already mined, as they have, some amazing projects, as you've seen tonight, for potential CMT treatments and cures. But we know for sure there are more gems out there, and we have to keep mining for them. The scientific team at the CMTRF has developed a phenomenal 3-year roadmap that will continue to focus on gene therapy development along with delivery vehicles. I can't stress how important the delivery side of this is. You can develop a drug that might work, but if you can't get it to the right place in the right time in the right proportion and not have an adverse outcome, it's not worth anything. So the delivery part of this is extraordinarily important. We're focusing on small molecules that address the core genetic mutations of CMT or the nerve degeneration downstream, we'll continue to assess those and prioritize those based on their ability to attract follow-on funding. Some of these projects don't just need initial sort of seed funding, they need ongoing funding to make it into clinical trials. You know, while CMT1A is important to you and your family, that's why you're here tonight, the CMT Research Foundation will continue to fund research to treat all types of CMT. We are not a CMT1 organization, CMT1A organization. I explained earlier why we started there, and it makes a ton of sense, but we're not exclusively a 1A organization. So I have to thank you as our supporters and our funders. Your confidence, your trust in us has fueled the progress that you've seen this evening. Candidly, your generosity and your trust truly humbles me. So we've got $2 million more to raise to complete our $10 million campaign, and we'd love your help. If we've moved you tonight, if we've stimulated you tonight, if you want to join our journey, if you haven't already, please do so. Every gift counts. $500, $1,000, $2,000, $50, whatever it might be. It's more important that you're in than the amount that you actually give. So thank you for that. And right now we have this very exciting announcement that we have a $100,000 challenge grant for all funds that get raised tonight. We have a very generous donor who will match all of the donations that are made tonight up to $100,000. And to learn more about that, please contact myself or Cleary or Susan or Anna Combs, our VP of Philanthropy. Anna, do you want to turn on and just say hello to everybody so they know who you are? Is she there? Okay, well, there's Anna. And please feel free to reach out to any of us. We'd love to talk to you some more. Now, the big thing I want to say also is we, as you know, host the Global Research Convention on CMT. This is the only one anywhere in the world. This is our, I'm going to say third year. I couldn't remember if it's 3 or 4 because of the COVID year. But we are doing it again this year in Cambridge at the Royal Sonesta, the same venue that we've done it the last year. If you want to come, please visit the CMTRF website to register for it. Okay, so we do have time for questions. So I see some questions out there. And with that, then I'll turn it over to my friend and colleague, Susan, who's going to ask these questions of our speakers. Susan.
Hi, Peter. Thank you. And before we get started with questions, we're going to pop up a poll here because we'd love to understand who's in our audience tonight. So you'll see that pop up on your screen. If you will please run through those answers as we begin to welcome our panelists back. So Peter, I see you, Dr. Quiroz and Dr. Loya, and Cleary, I want to make sure I see you there as well. Yep, you are awesome. Thank you. Okay, so our first question, this is for you, Dr. Quiroz. I'm getting lots of questions about what happens if there's not a trial site in my city or even in my country. Can I travel internationally to be part of the travel, or can I travel to different the different clinical sites to be part of the trial?
You need to contact the investigator or the clinical site where you want to attend. If that is across the state, you need to contact that investigators first and make the arrangements. So the answer is yes for the question about across the states. For international, it's a bit more complex, particularly in Europe. In North America, it's less so. Again, if you are in North America, you can contact, the trial is in English, and the materials will be in English only, not in Spanish. And you can contact, as of today, the investigator to whom you want is close to your place of residency, and arrangements can be made if all the conditions are met in that conversation. And again, this does not apply directly for Europe. It's not where we were discussing earlier today. It's not that clear that you can go from one country to the other one. There are issues about the ethics committees and legal, liabilities that people are concerned about. But the spirit of the study is that everybody is welcome. So we'll make all the efforts that if you have the interest to participate.
Thank you, Dr. Quiroz. And another question is, if the phase 2 trials at the current sites are positive, do you think you'll expand the trial sites for phase 3, or do you think you'll probably stay at the same? How do you make that decision, and when do you make that decision?
Yeah, that decision will be made at the finalization of the phase 2 study. There are many phases on research, and some studies and some drug development programs can be faster than others. We do not know the speed of this one. We have all the intention to make it as fast as possible. If the next study is a phase 3 study, will most likely require more patients to be involved in the investigative effort, and because of that, more sites to be included. And that may mean that we're expanding not only from the states that were not present in the US now, but also to Canada and perhaps Mexico, and in Europe, perhaps we will expand to Eastern Europe. So the answer to your question is yes, more patients will be required for the larger study, which provides the evidence that is sufficient for the FDA and the agencies in Europe to qualify a drug as efficacious and safe. And for that reason, more help will be welcome at that time.
Excellent. Thank you. Dr. Loya, we have a scientific question, and it seems like you're pretty good about breaking that down into knowledge or language that we can all understand. So we have a question about how the ASO actually gets released in the arrival of the target. So you've got your ASO into your antibody of the delivery truck. How does it actually get out of the delivery truck and into the cell and recode or do whatever it needs to do to help?
Yeah. That's a great question. So the way that the target protein that we're, targeting with our antibody, that has kind of, by nature, it actually is continuously being entered into and inserted into the cell. So it actually is very important, this particular protein, for transporting important kind of nutrients like iron into the cell. So we're kind of hijacking this particular natural biological system to essentially kind of hitchhike. I would say we're not hijacking, we're hitchhiking this particular system to insert our drugs into the cell. And once the drug is in the cell, then it can go into the specific kind of compartments where it can target the RNA that is being overexpressed or that is somehow kind of causing the disease. So that's how it would, in theory, work.
So it's kind of using the chemistry of the body already, and you're just using the system that the body already wants.
Exactly.
Got it. Thank you. Thank you. Dr. Agusquiza, we have a question for you, which is, besides targeting the mRNA, is there a way to lower the PMP22, maybe using epigenetics, to turn down the gene? So we're listening to mRNA studies and we're listening to neuromuscular junction. Are there other ways to turn down the PMP22?
Yes, definitely. So you can imagine that you could target, upstream of that mRNA, so more at the DNA level, or even downstream, at the protein level. So there are a lot of methods and approaches being explored on both ends there. In terms of epigenetics, I'm not aware of anyone exploring that right now, but it's a very interesting idea that I think is worth looking into.
Thank you. And one more question for you. This is a question we get a lot, which is, it seems like there's so much momentum for CMT1A, but if somebody has a rare type 2, or even a rare type 1, or a rare type X, or a recessive type, how does this progress? We like to say the rising tide lifts all boats. Can you talk a little bit about that?
Yeah, sure. So one of the big priorities right now at the foundation is figuring out these ways on how we can expand beyond 1A and leverage everything we've learned with the projects in 1A to other subtypes, as many subtypes as possible. I will say a lot of these projects that are focused on delivery of the genetic medicines, if you're able to find, how Carlos likes to say it, the delivery truck, then you can imagine that you can kind of plug in the right packaging or cargo specific to other subtypes. So I think all the work that we're doing at optimizing the delivery or really trying to find those optimized delivery approaches is going to really benefit a lot of different subtypes.
Excellent. Thank you. I think everyone in the CMT community can have hope, not only because of NMD Pharma's upcoming trial, but even because of 82VS's upcoming technology. We can kind of marry the two together and all keep working in the same direction for all types of CMT. Thank you. I think that's really reassuring to the entire community. So I'm taking a look at the clock, and it's 8 o'clock. And for those of you who've attended our webinars before, you know that we could go on and on for hours. What I'd like to do right now is invite Peter back to the screen to give us some closing remarks. And then for anybody else who would like to stay on, continue to ask some questions, we'll stay and hang out until about 8:15, answering some of these remaining questions. If you have more, pop them in the Q&A. I did see a question about republishing the poll, and we'll see if we can get that back up again for those of you who missed it. But Peter, if you'll bring us home, and then we'll open it back up for more questions.
Thank you, Susan. And we want a special thanks to our guest this evening. We're delighted you were able to join us this evening and motivated and stimulated by your remarks. It's, again, a very, very exciting time in CMT research overall. You know what? At the foundation, we're proud of what we've done, but we're just getting started. It's that simple. This is not something we're going to solve overnight. We know that. Folks have been at this for a long, long, long time, but the acceleration is obvious. The number of people in the community, the scientific community, who are engaged in this, the number of donors that we've been able to uncover and cultivate, the amount of money we've been able to raise are all telling us we're on the right track. We plan to stay on this track until we have therapies, until we have cures for all types of CMT. It's a tall order. I'm not going to kid you. But you know what? We're up for it. The team's up for it at the CMTRF. Our donor community has proven they're up for it with their stalwart support of us, and we're going to keep going. We're going to keep going until we take this disease off the front page. And so today's gift is tomorrow's cure. So if you're motivated and stimulated to help us, please give us a gift. And that's a gift of confidence as well as it is a gift of your financial resources. So thank you for that. And Susan, let's go back and answer some more questions.
All right. Thanks, Peter. And thanks to all of you for being here. For those who are ready to drop off, feel free to. Those who want to hang out for more questions, we'll stay until about 8:15. So Dr. Quiroz, there were a couple questions about after the phase 2, the people who participated in phase 2, will they be extended into phase 3, or will you be recruiting a whole new group of patients for phase 3?
Two questions there. Would it be an extension, which is usually called an open label extension, in addition to the end of the current trial? And the answer for that is no. And the reason is that this is very early days for this research. In general, the open label extensions are executed when you know that there is a positive profile of safety versus risk. And at this stage, we believe that that is so, and we are seeing that in the other diseases where we're trying the same medication, but we cannot guarantee that that is the case. So we need some sign of efficacy to be able to provide a longer treatment option for those who participate in the current trial. So that is in one hand. The other hand is that we are a very small pharmaceutical biotechnological company, and our resources are also limited. And therefore, the availability of medication that we have for NMD 670, we're using it all in the clinical trials to maximize the learnings that we can obtain in a controlled manner. You can also learn from open label extensions and for patients who have participated and donated their time and efforts, but at this stage, we're not able to do that. And the third consideration for that is something that is called the toxicological coverage, is that the agencies give you permission to dose for a certain number of days, according to what you have investigated, it is toxic or not in animals. And for us, the clock that we currently have is an approval for 4 weeks of treatment, the reason why we're doing 21 days. We already have information at hand for longer periods of treatment, and we're having those discussions with the FDA, so this may also change, but as of today, the treatment will be only to the clinical trial. To this clinical trial. The second question you had was if patients that participate in the phase 2 could participate in the phase 3. That is something that we have not discussed yet. I hope that we have that problem in terms of having a positive phase 2 study, and that will be a problem of good quality. Can we reincorporate the patients? We are perhaps 2 years earlier than that, so please let me pause to answer that question, and perhaps we'll meet in 2 years.
Sounds good. Thank you. I appreciate it. And Cleary, we have a question. Someone wants to know if they can make a donation and have it designated just for type 2s and not for type 1.
You can, absolutely. We will accept a restricted donation. You know, we really believe, and I feel like I should say this, that general donations unrestricted to the foundation indicate that donors trust that we will make the best decisions about the science. As Ryan was talking about before, so much has been learned by the projects that we funded in 1A, but we've also funded projects in type 2 and 1B and X. And really, we want to make sure that we can develop programs that provide treatments for all patients with CMT. So can you? Yes. We would encourage people to give more unrestricted funding as we go forward.
Yep. And
and also I'm happy to talk to anyone individually about that and more specifically.
That's right. Thank you, Cleary. Appreciate it. And Dr. Quiroz, and maybe even Carlos, this will be a question for you. You know, one of our attendees has a 15-month-old son with a different type of CMT than CMT1A. And, you know, Dr. Quiroz, you were talking a lot about the adult trial right now, phase 2. How do we think about clinical trials for children? What kind of feedback do you get from the FDA? And Carlos, you know, we'll have Dr. Quiroz answer first. And if you have a point of view to follow up, I'd love to hear your point of view as well.
Yeah, no, that is a great question. We do believe that this particular drug will be efficacious in all types of CMT, not only type 1 or 2, and that in all ages, not only adults and up to 70 years old. There is no reason why a 71 years old may not be able to benefit from the drug versus the younger one, and also in children. As commented with the duration of the trial, the agencies that regulate drug development, the FDA in the US and the EMA in Europe, they do have limitations on the age group that one could investigate in a clinical trial. And for that, you also need to demonstrate that in the preclinical species, the drug is safe to be administered in younger patients. So in general, those studies are conducted a bit later on when we have satisfied the primary question, which is this drug efficacious? So then to do that, you also need to agree with the agencies on how and when to do that. Now, there are certain cadence that needs to be followed. In fact, I can add to that the agencies obligate the companies to have a plan, even if the plan is to, in their minds, exclude patients, to have a plan in advance to investigate the potential of this drug in pediatric populations. Those are called the pediatric investigational plan and are mandatory in the case of Europe and also in the US. So in summary, we are planning to move this drug to younger populations. We cannot do it as of now because we have limitations on what the agencies allow us to do, but also because we first need to demonstrate that it's safe and efficacious in adults in this particular set of circumstances. And we are planning to move into pediatric populations in the later phases of development if this one is positive and efficacious.
Excellent. Thank you. And Dr. Loya, do you have anything to follow up with your experience?
I think that's perfectly said. So nothing necessarily to add there.
Okay. All right. Thank you. Thank you. Okay, so another question that we have is also for Dr. Quiroz. We knew you were going to get lots of questions about the trial design. And someone is asking, you know, you said that if they use an AFO daily, that they'll be excluded from the trial. Yeah. Does that mean that they'll be blocked from this you know, if they use them sometimes during the day, are they going to be able to participate in the trial? And is it more about the AFO, or is it more about the fact that they can't complete the 6-minute walk test without the AFO? That seems to be the concern. So if they could complete that 6-minute walk test without their AFO, then maybe they could participate.
Yeah, that's another great question. It depends on the definition. So this is something that you have to discuss with the physician that will be incorporating you in the study, if you qualify as using AFO or not. Using the AFO dailies, I will have the tendency to answer that that is somebody who does use AFOs. And the reasons for this are the following. The AFO users do walk a very different distance than the patients with no AFOs in the tests that we're measuring, and perform other tests of mobility also differently. That makes the data very difficult to be analyzed, and for that you need larger populations. So we believe that the question about the AFO use is something that needs to be done in a phase 3 study and not in a phase 2 study. I am aware that this is a novel, unfortunately, position, but we do believe that if we are restrictive now, we're increasing the probability of being successful in the future. We do not think that this drug will not help patients with AFO. There is no reason why it will help a patient with AFO versus a patient without AFO. But for the purpose of investigating the action in this very curated first clinical trial, we're excluding those, and the reasons were those two. I read the question, and they use the blog to participate sounds terrible in those terms. However, I think that one way of perceiving this is that the exclusion today will enable the inclusion tomorrow in the next clinical trial. The probabilities of being successful may be a little bit higher, and this is a hypothesis that we're testing.
Fantastic. Thank you. I love that the exclusions today enable the success of tomorrow. I think that's really important for all of us to understand that these studies really need to be designed so tightly, because we want it, if the drug works, we want to be able to show that the drug works, and we don't want the trial to fail because of poor design. And so that's why they can be so prohibitive. Thank you for that. All right, well, it's just about 8:15, and I think we've come to a stopping point with our questions. So I want to thank everybody who participated today. We had definitely a banner number of participants at the height of the webinar. Thank you to Dr. Quiroz from NMD Pharma. Thank you to Dr. Loya from 82VS and Alloy Therapeutics. We will be posting this webinar on our YouTube channel, and we'll send out a link. So feel free to come back and watch it. And if you do have any questions, you can follow up directly with me. I'm Susan at CMTRF.org. You can follow up with Cleary at CMTRF.org. We're all here for you. Follow up with us directly, and we'll get this on YouTube, and we hope to see you soon. Definitely come to that convention in September. It's the last weekend of September in Cambridge, Massachusetts. The folks from NMD Pharma and the folks from 82VS will be there in person, and you can meet them personally and ask any question you want. Thank you all for being here. Nice to see you, and have a great week.
Thanks, everyone.
everybody.
Thank you, everyone. Good night.